Hello everybody, this is Dr. Anjana Singh, Assistant Professor, Department of Botany, Government Model Degree College, Arnia, Bulandshahr. For this video lecture, I have selected this topic, and in this lecture, I will discuss about the morphology of one of the most important viruses, that is tobacco mosaic virus. So before going to the morphology, let's discuss about what are viruses. So this term is not new for us. So virus term is derived from the Latin word that means poison or venom or viscous fluid. According to Warden and Peary, viruses are described as some microscopic organisms which are generally less than 200 nanometer in size. They are obligate parasites and can pass through bacterial filters. In general, viruses are infectious agents of small size and simple in composition that can multiply only in living cells of animals, plants, or bacteria. The credit for the discovery of viruses goes to the Russian botanist D. Ivanovsky, who discovered the causal organism of tobacco mosaic disease in 1892. Later, after this, Bejernik in 1898 named these organisms Contagium vivum fluidum, means living infectious fluid. And since then, a number of viruses has been studied and discovered till date. These viruses show some specific features, like they are acellular, they are infectious, they consist of nucleic protein and a protein coat. Viruses reproduce inside the host cell, where they use host cell machinery for their replication. Viruses are obligate intracellular parasites, which means they survive only by using the resources of a host cells. Because viruses show both living and non-living characters, they are considered as transitional group between living and non-living. The morphology and the structure of viruses depend on the capsomeres. The capsomeres are the small proteinous subunit which form the capsid around the genetic material of any viruses. So viruses can be helical, polyhedral, or complex on the basis of this arrangement of the capsomeres. So let's see the morphology of tobacco mosaic virus. An electronic micrographic study shows that the TMV particles are rod shaped with 300 nanometer height and 18 nanometer diameter. They consist of nucleic acid, that is single stranded RNA, which is the genetic material of TMV, and this is surrounded by a protein coat that is known as capsid. The capsid of the TMV consists of about 2130 capsomeres, which are helically arranged around the central hollow tube called core, and this central core occupies 4 nanometer in diameter. There are 130 helices in total length of each rod. Inside this capsid, there is single stranded genetic material, single stranded RNA, which consists of 6500 nucleotide base pairs, and each turn The RNA contains 49 nucleotides. Each capsomere is linked with 3 nucleotides of RNA, which are also spirally coiled and form a helix. So in this picture you can see the capsomeres, or the small subunits of protein, spirally arranged around the coil of single stranded RNA, and it gives the TMV appearance like a rod shape or a cigarette like structure of 300 nanometer length. So in this case, the TMV genetic material is single stranded RNA, and it contains the genetic information and is responsible for the self-replication and the synthesis of virus specific proteins. So dear friends, this is all about the morphology of tobacco mosaic virus, which cause the tobacco mosaic disease in tobacco plant. In next video we'll see the bacteriophage structure. Thank you for watching this video.